Today we're going to look at starch digestion. For this experiment, we'll be using pancreatic amylase and examining the effect of amylase concentration on starch digestion. To look at the effect of concentration, we want to make 3 different concentrations of amylase. Start by labeling 3 test tubes, A, B, and C. Add 3 milliliters of water to each tube. Next, vortex your amylase solution to make sure it's well mixed. Then add 3 milliliters of your stock amylase solution to tube A. You'll have to use the 1 milliliter pipetter for this because the 5 ml pipetter won't fit. Now vortex tube A to make sure that it's well mixed. This is your first concentration, and it's now half the concentration of the stock amylase solution. Now take 3 milliliters from tube A and put it in tube B. This is your second concentration of amylase solution, and it will be 1/2 the concentration of tube A. Now mix tube B really well using the vortex. For your last concentration, take 3 milliliters from tube B and put it into tube C. This is your third and final concentration of amylase, and it's now 1/2 the concentration of tube B. Here's a diagram to outline what we've just done. Next, set up the experimental tubes. Label them 1, 2, and 3. Add 1.5 milliliters of pH 6.8 buffer to each tube. Starting with tube 1, add 2 milliliters of your amylase solution from tube A. Now add 2 milliliters of your amylase solution from tube B into tube 2. Now take 2 milliliters of amylase solution from tube C and add it to tube 3. For this demonstration, we'll use tube 1. Set up your test plates with 1 drop of the IKI solution in each well. Vortex your starch solution to make sure it's well mixed. Then add 1 milliliter of the starch solution to tube 1. As soon as you add it, mix it and start your timer. Use a clean transfer pipette and add 1 drop of your experimental solution to 1 well plate. Do this every 10 seconds until the solution no longer changes color. The presence of starch in the solution will cause the IKI solution to turn a dark blue color. When all the starch has been digested, the IKI solution will remain an amber yellow color. When there's no further change in color, stop the timer and record the time taken for starch digestion. Repeat this procedure for the remaining tubes, but use the time intervals indicated in your lab outline.